Okay, welcome to Level 1 Science Online with Henny van Vuren.
Good morning, Year 11s. Welcome to one of our last lessons before the exams. I really hope that this will help you to do some revision. So this is the 2019 past paper for genetic variation and change. So what I want you to do is to please have some paper ready and then write the answers down. As we go through the question, I need you to pause the video and then write down your answer, and then please can you email me the photos of your completion. Yeah, so please pause it before I show the answer and then we move on. So question 1 was on DNA structure, and you can see over here it says label the blank bases A, G, T or C in the diagram of the DNA shown below, and we know that it is at the Gold Coast, okay? And that is how they always mix. Okay, so the base pairs, that is called the complementary base pairing rule, okay? And you need to remember that because you're going to use that later. Okay, so can I ask you to quickly stop the video and then just fill in the blanks for me? So pause the video and then fill in the blanks. Cool, welcome back. Let's check how you did. So obviously A goes to T, so G goes to C, T goes to A, and C goes to G. Wow, that was really difficult, wasn't it? I wonder what the model answer says. The model answer says any A to T or G to C correct base pairing, you get 1 achieved point. Wow, that is not difficult. You agree? So if you had any of them correct, you would have gotten an achieved over here. So I am sure that you can give yourself an achieved now. Woo! Good job. So some adults can digest milk, but the majority of adults in the world, believe it or not, cannot. The ability to digest milk was caused by a mutation. So firstly, what is a mutation? And the second part of the question is, explain how a mutation can give adults the ability to digest milk. You should include the terms DNA, gene, allele, phenotype, and mutation in your answer. Okay, so quickly pause the video and then write down your answer and then come back and let's see how we did. Welcome back. So yeah, so a mutation is a permanent change in the DNA sequence of a gene, and it leads to a new trait. Nice. And then explain how a mutation can give adults the ability to digest milk. In your answer, you should include the terms DNA, gene, allele, phenotype, and mutation. Now, like all of these, we've just given the definition of mutation upstairs, but in biology, when we answer any questions, we give the definition first, or as we start talking about, we give the definition. So, if you look at this, the first thing they want us to talk about is DNA. So, DNA is the genetic info of our bodies. Okay, and a gene is a section of DNA that codes for a specific trait or protein. Alrighty. Now, an allele is an alternative form. Of the same gene. And a phenotype is the physical feature. Remember the F alliteration? The physical feature displayed due to the genotype. Okay, so now We've made all the definitions there, and that is all just for achieved level answers, okay? But now we need to go back and say, so the DNA is the genetic code of our bodies. A gene is a section of the DNA that codes for a specific trait or protein. An allele is an alternative form of that, and a phenotype is the physical feature displayed. The only way that we can get a new allele, so, is by a mutation. Okay, so only way new allele is mutation. And that is a change in the base sequence. Okay, so, and a mutation could have occurred that allowed humans or adults to keep on digesting the lactose in milk, and therefore a new allele was formed, and that was called, well, we call it lactase persistence, but just the ability. So a random mutation, that's interesting, now I'm a black pen again, a random mutation occurred, giving us the ability to digest milk, and that is a new allele. Okay, now it specifically says the next part of it. Well, let's go and look at our model answer. So a mutation is a change in the order of the DNA bases that can cause a new allele. Yay, we got that. So if you said a mutation causes a new allele, or you said a mutation is a change in the DNA, you get another A. I think we did that. Now the next part is a excellence question. So a mutation is a change in the order of the DNA bases that cause a new allele. We said that. A DNA carries genetic information as a base code. We did say that. A gene is a sequence of DNA that codes for one trait or protein, in this case digesting milk. An allele is a different form or version or expression of the same gene. A mutation is a change in the order of the DNA bases that causes a new allele. The new allele codes for the phenotype ability to digest milk as an adult. Okay, we wrote that down too. So we defined DNA as genetic material. We said that a gene is a section of DNA. Can you see there's an A there for you, an A there for you? Allele is a different form of the same allele. So if you're only aiming for achieved, you have now already gotten an A5. You've already had 5 opportunities for achieved. Okay? And all you had to do was memorize your definitions. So it's not a lot of work if you're just aiming for achieved. And then to take it one step further, just showing some understanding is merit, and you will do great with that. So define phenotype as the physical expression of the genotype, the ability to digest milk. Yep, we did that. Beautiful. So for merit, if you wrote a mutation is a change in the order of the DNA bases that create a new allele, causing a and new phenotype or protein. Yes, we said that. So that's merit level. And then for excellence, there's 2 excellence points here. So the first one said a mutation is a change in the order of DNA bases that create a new allele. The new allele codes for the ability to digest milk. Yep, we did that. But remember, you also had to do all the other things. So if you didn't define them like we will learn that we should always do, then you wouldn't have gotten that E. Okay, so we just now go and look, because this one goes with the next section. Okay, then explain how a mutation can be passed on to the next generation. So, mutations happening in body cells cannot be passed on. Remember, body cells, also called somatic cells, are not inherited. Now, also check any words in here that we haven't defined yet. No. Okay. And passed on means fertilization. So, mutations in somatic cells are not inherited. Only gametic mutations can be passed on or inherited. That means that during the production of the sperm and the egg, the sperm and egg, the sperm or egg had a mutation, and that allowed for the new allele of milk digestion, and that would have been then carried in the sperm of the egg. Then, what's more important is that the gamete with the mutation, so the mutated gamete, had to be fertilized. to create a new zygote. That carried this new allele. Okay, so only gametic mutations can be inherited. Yeah, I think that covers everything. Let's have a look at the model answer. So, a mutation coding for the ability to digest milk as an adult can be passed down in the DNA from either parent when sperm and egg gametes fuse. Described as passing on in the sperm and the egg. We did that, so we got an A. Can be passed down in the DNA when sperm and egg fuse. Fertilization. Yes, we talked about that. And then DNA can be passed on in the gametes when the sperm and egg fused, forming a zygote. Yep, or you use the word fertilization. And a comparison of the somatic cells which cannot be passed on. We did that. Yay! Okay, so that was quick. Question 1. We have an E8. I'm really happy to tell you. So, let's have a look at the next question. Oh yay, Punnett squares. Question 2, cystic fibrosis. Cystic fibrosis is genetically, that's a big word, inherited condition. It can be traced through a family as shown in the pedigree chart. Cystic fibrosis allele T is recessive to the normal or unaffected allele. Very important. So, here's our key. Affected is colored and unaffected is not colored in. So if you look at it, complete the Punnett square for the cross between individual 1 and individual 2. That is interesting. Complete the Punnett square for the cross between individual 1 and individual 2. Now let's look at this. So if you are normal, you have definitely 1 at least capital T. Do you agree? Now, remember when we're working with T's, do not make a big T like that and a little t like that, or even like that. A little t needs to have the curly bit, otherwise the examiner won't be able to notice the difference, okay? Don't use letters that look the same in capital and non-caps. So, if they have children that show a recessive trait, because remember, if you display the recessive trait, it means that you have to be little t, little t. You agree? That meant that mom had to give a little t and dad had to give a little t. So both mom and dad have a little t, and both mom and dad, because they displayed the normal phenotype, had to have a big T. Now, we can immediately say what these are, but we cannot say what number 3 and number 6 is, because dad could have given a capital T and mom could have given a capital T or a little t. So we actually don't know, but we do know that they contain at least one capital, or the dominant unaffected. Okay, so complete the Punnett square for the cross between individuals 1 and 2. So we just said that individuals 1 and 2 had to be heterozygous. So heterozygous, which means when they make sperm in this case, then he's going to give one big T and one little t. The mom is also heterozygous, so when she makes eggs, it's going to be a big T and a little t. And this person will be homozygous normal, and then this person will be a carrier of the cystic fibrosis gene, but they are still displaying the normal phenotype. This person here will be the same carrier, and over here we have somebody who then suffers from cystic fibrosis. So it shows us that work out the genotypes of the following 3 individuals. Now we already did that, so individual 5 was little t, and then 1 and 2 over there is big T, little t, big T, little t. Okay, so explain any difference in the expected phenotypes of the offspring ratio to the actual phenotypes of the offspring ratio between the cross with individual 1 and individual 2? In your answer, you should refer to the expected and actual phenotype ratios. So expected and actual phenotype ratio. So in your answer, you're going to have to tell me what is a phenotype. Remember, because we need to define that. And then also what is an expected phenotype ratio. So let's look at our expected phenotype ratio. So an expected phenotype ratio would be 1 normal to 2 carriers to 1 cystic fibrosis sufferer. That means that 25% of the offspring, because there's 4, 25% should have cystic fibrosis and 75% normal. Okay, if you just look at the phenotype. But when we then look at the actual offspring, we can see that 50% of them were cystic fibrosis. Now, how is that possible? Please answer the question for us. Welcome back. So yeah, the expected phenotype ratio was 75% normal and 25% cystic fibrosis. But the actual phenotype was 50% normal and 50% cystic fibrosis, or 1 to 1, and in this case 3 to 1. Okay, so in your answer you should refer to the expected. So explain the difference in expected phenotype of the offspring and actual phenotype. So the first thing we do, we define a phenotype again. So a phenotype is the physical feature. And in this case, it is either normal or affected. Okay, now a Punnett square refers to every fertilization has a 75 to 25%. It doesn't look at the total kids. Okay, so every time there's a fertilization between these two individuals, there's a 25% chance of producing an offspring with cystic fibrosis. Okay, and so let me say, even though the phenotype ratio expected is 75% normal to 25% CF, this is for every fertilization. Okay, and does not refer to all offspring or combined offspring. So our actual phenotype ended up being 50%. Holy moly. So 50% normal, because they had a 25% chance every time there's a fertilization. Okay, so that should explain it. Let's go and look at the model answer. Okay, so 2 correct genotypes in the same row or column. So basically what they said here is that if you have that one correct and that one correct, or that one correct and this one correct, or this one correct and this one correct, then you got an A. So you could have actually made a mistake and had the parents both capital. Oh no, that wouldn't work. But let's say you had a little. That would have meant that you got these two correct, and consequently you still get your A. Now, any correct genotype can be taken from the Punnett. Holy moly. So you get another A if you had any correct genotype. If you said individual 1, individual 2, or individual 5. You just had to get one of them for another. Then here's our answer. The expected genotype ratio was 3 to 1 unaffected to cystic fibrosis respectively. We can also, as markers, accept 75% unaffected to 25% cystic fibrosis, or 3/4 and 1/4 affected. The actual ratio is 2 to 2, or 1 to 1, or 50-50. Individual 1 and individual 2 have more cystic fibrosis than expected. No, have more offspring with cystic fibrosis than expected. This is because each offspring is the product of random fertilization. I didn't say that. Each offspring is unaffected by the previous outcome because it is during fertilization. The Punnett square is for fertilization, and it is a random event, random fertilization. Since individual 1 and 2 are both heterozygous, each individual have 50% chance of inheriting either the allele from each parent. It's an interesting statement to make. With a larger number of offspring, we would expect a ratio closer to 3 to 1. Okay, so you wrote the expected ratio, we wrote the actual ratio. Described random fertilization. I did not do that. Describe the Punnett square as only showing possible predicted outcomes. We did that. And we mentioned small sample size. I didn't do that. See why it's important to do past papers. Explain the actual ratio of cystic fibrosis is different than expected, and difference depends on chance or random. So see there, I dropped off. I can't get this M. Explain the affected predicted ratio is the same for each offspring, as each fertilization is a random or independent event. I did say that each one is an independent event, so we can get an M for that. Since individual 1 and 2 are both heterozygous, each offspring has a 25% chance of having cystic fibrosis by inheriting a recessive allele from both parents. I don't think we actually said that in so many words, but we obviously implied that. So I'm not going to do that great in this one. If I was really lucky, the marker could give me an M there. But the most I would probably get here is an M6. So explains the actual ratio of cystic fibrosis is different than expected because each offspring is a result of random fertilization, and each offspring is unaffected by the previous outcomes. Please remember these words. With a larger number of offspring, we would expect a ratio very close to 3 to 1. So with increased numbers, so if you were breeding rats or if you were breeding guinea pigs and you monitor all the offspring, eventually you will see a ratio that is closer to 3 to 1. But because in this case there's only 4 children, hence it's not a big enough sample size to get that, and because everyone is unaffected by the previous one. So that's why we get the 3 to 1 ratio and not, oh sorry, that's why we get the 1 to 1 ratio and not the 3 to 1 ratio. Okay, here's question 3. So we're going on to question 3 over here. Ooh, Cody dieback. Remember, I did my honors degree on Phytophthora cinnamomi, just for interest sake. Phytophthora cinnamomi. Cinnamomi. Okay. It is phyto means plant, phora means killer, and they first found it on the cinnamon tree because cinnamon is the bark of the cinnamon tree. Now, this organism here is killing off our kauri in New Zealand, and I've told you about that. It's got a spore, a motile spore that can actually swim when it's wet, and that's why if you have it on your boots and even if you don't walk that close to the kauri, it might still make its way to the kauri roots. Anyway, so kauri dieback disease damages the tissue that carries the nutrients within the kauri tree. This means some trees survive and others starve to death. Now, if you remember back to year 9 when you learn about the xylem and the phloem and how do you get the actual water all the way to the leaves, and how do you get the sugar produced in photosynthesis in leaves to go down? Those are the basically like vessels, almost like arteries and veins, and the pathogen in this case, the fungus, grows inside those tubes and it blocks the tubes, and hence you will see literally half of the tree dies because it's not getting any water or it's not getting any nutrients down to the roots. But it starts in the roots and then grows up. Okay, so enough of a sidetrack. The kauri dieback disease damages the tissue that carries the nutrients within the kauri tree. This means that some trees survive and others starve to death. Describe genetic variation in kauri trees. And then question B is, explain how the sexual reproduction of kauri trees cause genetic variation, and how this could lead to increased survival of the species when faced with kauri dieback. In your answer, you should consider the gamete formation or meiosis and fertilization. Okay, and I want to add here random fertilization. How sexual reproduction leads to variation of the population, and the link between genetic variation and survival of the kauri. So to give you some guidance here before you start answering, so you're going to have to explain meiosis for us, explain fertilization, and also describe and define them, explain sexual reproduction as the sperm and the egg coming together and randomly, and how that then causes variation. Okay, so, and we talked about the few steps in meiosis that causes variation. So remember, it is random assortment. So random assortment, next one was crossing over, and then segregation and random fertilization. Okay, so just 4 steps. So can you please pause and then go and answer this for me? Okay, welcome back. Genetic variation is when there is high genetic variation, in other words, lots of different alleles for the same genes. So if you think about hair color, all the different variations of hair color, or what we want in this case, we want to say that lots of different alleles for the same genes, and this would be fungal disease resistance. And just like in your family, your mom or your dad might get a cold and they might be almost dead or feel like they're dying, whereas you get the same cold from them and you're not sick at all. You're just a little bit sniffles. So it's the same way. Genetically, you are simply more resistant to that specific pathogen. Okay, so genetic variation is the amount of different alleles for the same genes in a population. Okay, and the higher the genetic variation, the better for the species survival. So increased genetic variation leads to species survival. Okay, and that is what why I made you watch that video about the bananas. All the bananas are clones, and that means they have a very, very, very shallow gene pool, which means at the moment we have, what is it, banana blight, or was it banana rust? I think it was banana blight that is killing off huge patches. So they also have to isolate them, just like we have to isolate with COVID. Now, explain how sexual reproduction of kauri trees cause genetic variation and how this could lead to increased survival of a species when faced with kauri dieback disease. In your answer, you should consider the process of gamete formation. Meiosis. So, meiosis is the process of gamete formation. This is probably a little redundant, but okay, and the first step is that the homologous pairs of chromosomes, and what is a homologous pair of chromosomes? Remember, you have 2 chromosome 1s, 2 chromosome 2s, 2 chromosome 16s, 2 chromosome 18s, 2 chromosome 23s. Those are all homologous pairs. It means they carry the same genes, but the different alleles will determine your phenotype. So homologous pairs of chromosomes, they pair up along the midline of the cell. Because at this stage, I'm saying cell and not nucleus, because the nucleus, the membrane has actually been removed. Okay, so they pair up along the midline of the cell, and this is where random assortment comes in. It is randomly assorted to the left and right of the midline. So in other words, whether the chromosome is from maternal or paternal origin. So remember, we're talking about if you are making eggs, then half your one chromosome came from your dad and your other chromosome came from your mom. And whether this is from your mom or dad, it goes left or right randomly. So whether maternal or paternal origin, left and right is random. And this, when they segregate, creates lots of variation. Because now you have a combination of mom chromosome, dad chromosome, mom chromosome, dad chromosome, and so on. So in segregation, this gives lots of variation. But before variation, however, before we get to segregation, they do that funky crossing over thing. So the homologous pairs, as they are lined up, the non-sister chromatids cross over and swap sections of DNA. Okay, and this leads to 4 unique chromatids. And if you want to, you can draw a little picture here. You can say, so let's say you have the 1 chromosome there. That is a chromatid, remember, but the whole chromosome is still joined, so we talk about 1 chromosome. And then it has swapped over because this guy, okay, so these are 2 sister chromatids of 1 chromosome. But the 2 non-sister chromatids, that means this one and this one, they cross over and swap sections. So now you have 4 unique chromatids. Okay, I'll go back to our question. So homologous pairs are segregated, and then sister chromatids are segregated. So remember, segregation means a little spindle fiber comes and pulls this one this way and that one this way, and then the cell divides. Okay, and after the cell divides, now you have 2 cells, and then we break this one apart and we split the other sister chromatids. Alrighty. Okay, so what do we get at the end of meiosis? So homologous pairs segregated, then sister chromatids segregated. And that leads to 4 unique haploid, which means half daughter cells, which equals gametes. Now, whichever one is fertilized is completely random. So random fertilization says that a random sperm and a random egg, so the fertilization is random between the male and the female gamete, because we cannot predict which one is going to fertilize. Okay, so in the case of the cari, we don't know which piece of pollen will fall on which little flower. Okay, so fertilization, I'm just going to say here random, but you need to elaborate more. And because of that, you can pass on the different alleles. Okay, and because when we then pass on and we create these new combinations, some organisms, because remember our question said now, link between the genetic variation and the survival. How sexual reproduction leads to variation, we just did that. And now link between genetic variation and survival. Now because we have these unique haploid cells and gametes, and potentially different alleles formed due to mutations, and they are then passed on and forming new gametes and new zygotes, and we might have zygotes that have an increased immunity because of, so increased immunity in Cody because of the random fertilization and the variation occurring in haploid daughter cell productions. Okay, so I hope that makes sense. Let's go and read the model answer. I'm trying to write as fast as I can, that's why I'm not writing full sentences, otherwise you're going to get so bored. So, define genetic variation as the differences in DNA or genes and alleles. Yes, we did that. So define it as the differences in the number of alleles specifically. Okay, so we got an A. Gamete formation is by meiosis, random assortment, segregation, crossing over of the chromosomes in meiosis. A description of meiosis produce gametes or sex cells that have half or haploid the normal number of chromosomes as body cells. Yep, we did that. So define meiosis as the process of having halving the chromosome number. Yep. And define a gamete as a sex cell, sperm or egg with half the number of chromosomes. We did that too. Okay, how random assortment of DNA and chromosomes during meiosis create variations. So we did that. Explain fertilization and how the random nature of two parents create genetic unique. We did that with, I didn't go deep enough into that, I would say. We need to talk about when these two haploids come together, they mix their unique DNA, and as that unique DNA forms a zygote, the zygote is then completely unique with a new combination of alleles. Okay, and then going back to here, the process of fertilization, random male and female gametes join, each with a unique DNA producing genetically unique zygote offspring. The role of sexual reproduction produce new combination of alleles, thus genetic variation between them. And then explain, because this is linking back to the Cowdy. If you don't link back to your example, you will not get your E. The advantage of genetic variation to a species that may enable some individuals to survive Cowdy dieback, to produce passing on favorable alleles or genes to the next generation. Over many generations, this genetic advantage will rise in the population, allowing survival of. So that's a little bit of natural selection over there. So define sexual reproduction as the mixing of DNA of the two gametes. Yep. Define fertilization as sperm and egg joining. Yep. And remember, this one is an OR. So describe the advantage of sexual reproduction to produce variation, or describe an advantage of sexual reproduction. Okay, so there you go. And then for the next merit point, individuals are genetically varied and some survive Cowdy dieback disease and reproduce more. And then lastly, your two excellence points. Explain the role of sexual reproduction causing genetic variation and how genetic variation in a population leads some individuals to survive Cowdy dieback and reproduce, passing on favorable alleles to the next generation. We did that. However, we didn't do this. Over many generations, the favorable genetic advantage alleles will arise in number in the population, allowing the survival of the Cowdy species. So we did not talk about natural selection. I hope that you did, but I know I did not. So the maximum I'm going to get for this one is an E7, and then I can add my grades up. So it was a 7 plus a 6 plus an 8. 7 and 6 is 13. 13 plus 8 is 21. Yay! I got an overall 21, which I'm relatively happy with. Okay, so there you go, guys. I hope that helps, and yeah, please remember to prepare during the holidays. The exam starts in the first week, so we need to make sure that we get that done. Thank you so much. Look after yourself. Keep well. Bye.